And this is just a review. Remember that the DNA backbone forming a right-handed helix. Remember that the DNA strand has a polarity and a directionality that go from 3 prime to 5 prime, here 5 prime to 3 prime. So remember all those things, and remember that they are anti-parallel. So all what it means, so that one go in one direction while the other one go in the opposite direction. So just remember all those things. And again, this is just an introduction to what you have studied in high school and what you have studied also in genetic revolution. So when we talk about replication, so remember that, and this is the schematic that they show in your textbook. It's very similar to what it happened in what we study in the Campbell biology. So you have a parental duplex, and you have to unwind it to create a template strands and a replica strands. So here, just to remind you again, key to study DNA replication is the presence of these template strands and the complementarity. So I know you remember this. So here you have your DNA, you use heat or you use an enzyme and you can open them. And when you open them, you will have a template, and you will also have a daughter strand that it will be created by the complementarity of the DNA of the base pair. So remember, since the two DNA strands are complementary, each one act as a template for building a new strand during replication. And in DNA replication, the molecules unwind, as I show you here. And the two new daughter strands are built based on the base pairing rules. So if you remember this from cells and energy and genetic revolution, there were at least 3 alternative models explaining DNA replication. We have the conservative model, in which one you have the 2 strands, and those 2 strands, the old strands will join with only with all strands, while the new strands, so let's put it here with light blue, will not mix with the old strands, so you will have to have they will be part of a new strands. Yeah, so all strands will rejoin with all strands, new strands will rejoin with new strands. Another model, it was the dispersive model, and here you have a combination of both. So you see that the old strands and the new strands will be mixed in small pieces in each generation. So remember, conservative model, the 2 parent strands rejoin and 2 daughter strands will rejoin. While in the dispersive model, each strand is a mix of an old and a new strand. Those 2 models they were found that they were incorrect. Actually, it's a semi-conservative model, as you. So the correct model was the semi-conservative model, and if you see here, you have an old strand that will rejoin with a new strand. So here you have an old strand and an old strand, and here you have an old strand, and it will rejoin with a new strand here, and with a new strand here, and the same thing here. So remember, Watson and Crick semi-conservative model of replication predict when the double helix replicates. And remember, each daughter molecule will have one parental strand and one newly made strand. And they were correct. And I'm going to show you a video that explains how Meselson created an experiment to test that. And we're going to just, in this introduction, talk very briefly about that. But later in the class, in the course, we will have, in the middle of the course, we will have a chapter that focus on this and explain it in a lot of detail.
The Meselson and Stahl experiment provides evidence for semiconservative replication of the DNA molecule, where the 2 parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells therefore contained heavy N15 nitrogen. The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected. The DNA was then extracted and dissolved in a solution of cesium chloride. The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube. DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moved to a position in the gradient determined by its density. DNA containing N15 is denser than that containing N14, so it sank to a lower position in the cesium gradient. After one generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only one strand of each duplex contained N15. After two generations in N14 medium, two bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Meselson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
Okay, I think this was very clear, and as I said before, we will talk in more detail about that in the middle of the course. So now we are moving to something that we didn't study in genetic evolution. Highly recommended that you go to the textbook and read this section. Yeah, that is the relationship between gene and enzymes. Why? Because, okay, we know that genes are important. We know that at this point that DNA, that is in the DNA, the genes, but we don't know exactly what they are doing. And these experiments that I will show you, they show that somebody that he was working with fruit flies, that were many of the experiments. If you remember, Morgan found very interesting experiments in the eye color, and this person went beyond those experiments. And but to do that, he had to use even a simpler organisms. That it was a fungus, and that this new model organism allow us to make a connection between genes and enzymes. That genes are doing something that is controlling the production of proteins, and in this case, the control of enzymes. So there was a genetic control of biochemical events. So let's talk a little bit about that, and I will spend a little more time. Highly recommended that you study this, that you read this in the textbook, that this is possibly you have never heard about this. So first thing that you need to study is these guys, beetle and totem. Use this organism called Neurospora crassa, that is a fungus that is very easy to grow, grows in bread, to study this question that there was a relationship between gene and enzymes. So now, and you will see that, so for example, you will have a gene, and this gene will encode, in this case, a phenotype that it will be growth of this fungus. Why? Because those fungus need some enzymes. So when you have all the enzymes active, they will be able to grow. If they don't have this enzyme active, you will have a problem. And how we test these ideas, and we will discuss that later. You will create mutations. So you just same thing that happened with fruit flies, and that you will see in the lab. If you have a mutation, you will have an enzyme that is not working anymore. So the experimental approach now is called the genetic analysis, and it leads to this hypothesis that is the one gene, one enzyme hypothesis. And this one gene and one enzyme hypothesis, it was great as a hypothesis. It showed that there was a relationship between genotype and phenotype, as I will talk later, and we talk in the genetic revolution. Although in some cases the relationship between genotype and phenotype is simple. Not always we are going to find a clear and simple relationship, but it will be more complex. But was a great beginning to study the relationship between genotype and phenotype. So remember, as I said in the previous slide, that was a great beginning to study the relationship between gene and phenotypes. And this organism, Neurospora crassa, that sounds very weird. It's actually a common bread mold, and here you find a picture of this fungus. And this fungus has nucleus with 7 chromosomes, and if you see, each one of these green spots show that each filament is multinucleotide. So it has a multinucleates. And something that is very important for you to understand these experiments is that they create this minimal medium that contain the minimal resources required for growth. So it's a combination between salts, sugars, and vitamin. Okay, now I'm going to show you this video that explains very clearly some of the basis of this experiment.
One of the geneticists who studied fruit flies alongside Morgan was George Beadle. Working in Paris in 1935, Beadle detected evidence that the inherited trait of eye color in Drosophila might be the result of genetically based chemical reactions. Beadle pursued his investigations at Stanford University with colleague Edward Tatum. For their experiments, Beadle and Tatum selected another simple organism, Neurospora crassa, bread mold. They chose mold because it was easy to grow with simple nutritional needs, bread, air, and water. Neurospora also had a single set of chromosomes, which allowed the researchers to observe genetic changes easily. Knowing that X-rays damage the chromosomes, Beadle and Tatum irradiated the mold, which caused mutations in the genes of its spores. These mutant genes were unable to produce the nutrients necessary for the mold to grow. However, when they added the nutrients, some of the spores began to germinate. For Beadle and Tatum, this was a breakthrough moment. They realized that the irradiated spores failed to produce the essential nutrients because their genes were defective. This was significant. It meant that genes were responsible for more than just passing inherited traits from one generation to the next. They also directed the production of enzymes that the mold depended on for its survival. Beadle and Tatum's breakthrough is known as the one gene, one enzyme hypothesis. Lactose intolerance is an example of a human metabolic condition caused by a single missing enzyme. Missing because some people have inherited a gene that fails to produce lactase and are unable to digest the sugar in dairy products. This disorder can be easily treated by taking tablets containing the missing enzyme. Thanks to Beadle and Tatum, we gained a fundamental understanding of what genes do, and the stage was set for a new generation of remarkable genetic discoveries.
Okay, I think this video explained very clear the importance of this experiment, and as I said before, we are still using the same technique that with different organisms and with different purposes to study this type of questions. So let me move into and explain a little bit more about that. So this technique that they describe using, okay, let me take this green again, this color again, generating mutants strain in this fungus. They use, this is a technique, use mutant screens, and they are still used, for example, to study cancer. We just take a lot of mutation and we study not only one gene but the combination of several genes and see if they produce metastases. So, and we use simple organisms like fruit flies to then focus on the most on the most interesting genes, to then move into genes like into other more organisms like mice, to then move slowly into more applications into humans. So the same technique of using multiple mutations to screens is still used. Um, and actually, many of my friends, they were looking for new mutation affecting a particular effect, and so it's something that even now is still used. Uh, mm, and mutations, mutant screens sometimes isolate different mutation in the same gene. So that is very important. Why? Because remember that a mutation is just a change in the DNA. So as we will describe in the following slides. They could be just point mutation. You can have you are missing maybe multiple parts of the DNA. So a mutation, it could be so many things. It's like when you hit a punch a TV and you see, oh my God, the TV is not working. This is more or less what, at least in the beginning, when we use mutations through irradiation, we just are creating multiple changes at the same time. So, and this is related to the question that they solve. So, key idea here is that mutant spores can grow in a complete medium, but not in a minimal medium. What this means? So you have, or fungus will have multiple pathways for growth. Yeah. So when you have a complete media, if you have a mutation here, they will start using another pathway to grow. However, in a minimal media, you will be using only the essentials for growth, and if you have here a mutation, there will be no growth. So it will be an easier way to test if those genes are involved in those enzymatic reactions. So um, knowing that, what they did is they took the, as a control, the complete media, and they compare it with the minimal media, and they found that of course they will have many of the mutations are going to grow in both. Yeah. So if you have a mutation here, it's not going to have an effect. If you're going to have a mutation here, it's not going to have an effect. If you have a mutation here, it's not going to have an effect. The only ones that are going to be have an effect, it will be the ones that are involved in this pathway. Yeah. So most of them will be discarded because they grow in both. However, few of them, a few strains fail to grow in a minimal media, and those are the ones that are studied further. So the ones that are here that they cause no growth are the ones that we're going to study. The next step is to study that particular mutant in more detail, and they give them more a mixture of nutrients to see which is the nutrient that are actually missing. So in all of these ones, so in addition to have the minimal medium, so you say minimal medium, you start adding, for example, vitamins and see if vitamins are the problem. So look, vitamin was not the problem. What about if nucleic acid precursors? It was not the problem. Look what happened here. Amino acids, when you add minimal medium plus amino acids, they grow. So it seems that the mutation related in this case was to a particular amino acids. So remember, mutant strains are tested on minimal medium plus supplemented with mixture of additional nutrients, and the mutant strain here require amino acids to grow. So that was a key step. So to study in more detail the mutation. And then you can still study in more detail which specific amino acid is the one that is studied, they are needing. So it's leucine, valine, glycine, etc., etc. And for example, here, what they are showing is that this one in particular is the one that is used. So again, here we are going from a very general to a more specific. It was not only amino acid, that it was this specific amino acid, the one that is required. Uh, so this mutant strain in particular required the arginine to grow. Now I want that the key component here, or the main idea here, is not the names. I just wanted you to remember that when you don't have this amino acid, there is an absence of an amino acid. Now you can also study the precursors to study the steps of the reaction. So, for example, you have one component of the reaction here and another component of the reaction here, and you can start studying which one go first and which one go second based on the results. So, for example, in the urea cycle, you have to produce these amino acids. You will need these components, yeah? But if you don't have one of these, maybe you will not have the next step. So let me show you in more detail. So you have a complementation text to bring two mutant genes together in the same cell or organism. So here, in addition to the minimal medium, you have one of the precursors, and here, for example, there is no growth. Well, there is growth in the other two. So that can give us information to know which one is the first one, then the second one, and then the third one in a metabolic pathway. So let me show you that with an example in more detail. Okay, so now let's explain this in more detail. And now to do that, I have to tell you that what they did is the experiment of the growth. We start with the minimal medium, and you will find growth every time. However, when you have these three mutants, you don't find growth with the minimal media. However, when you add a precursor 1 or 2 or the amino acids, in the wild type everything grows. However, in the other ones, if you see the other ones, sometimes there is growth and sometimes not. And depending on your on the growth, you can dissect which one was which one is the metabolic pathway, the order of the metabolic pathway, which enzyme go first, yeah, which enzyme go second, and which enzyme go third. And in the case of the, the wild type, as all of the pathways and all the genes are working, so you go from the precursor 1 to the precursor 2, and you will be able to produce the protein at the end. However, as I said before, in this one, definitely in the minimal media, you are not going to be able to produce it, and in the other ones, depending on the growth, you can dissect the metabolic pathway. So let me explain this in more detail in the next slide. And one way to explain this, hopefully, is clear. Imagine that you want to go to Vancouver to Toronto. So if you want to go from Vancouver, maybe you will have to go then to Calgary, and you have to go to Winnipeg, and then you will arrive to Toronto. Yeah, you're going to drive, and you have a lot of time, free time, and you want to see all the different parts of Canada. So imagine that when you're driving this, you're going in your car, and there is a snowstorm or there something happened, and if the street is not working very well, that there is a problem in the highway, and if there is a problem in Calgary, you cannot go. Not only Calgary, you will not be able to go to Winnipeg, and you are not going to be able to go to Toronto. So what was the problem? That there was a problem in the highway in between Vancouver and Calgary. So the problem was here. Yeah. So the same thing happened with the proteins. Depend on the gene and the location, you will be able to go to the next steps. Yeah. And this is the key idea to understand what they did in this in this step. So here, so for example, when you are not going to have growth in the minimal media, yeah, but you have growth in the other three. Why? Because you have the precursor. If you have this precursor and you have the enzyme B, you can move to the next one. And if you have the enzyme C, you easily can go to the next one. So this is why you find growth when you have this precursor. You can definitely find, you can grow in the other media. However, here, when you have this, you don't find growth in this one, in the precursor number 1, might be because the enzyme B is not working, because you can have this precursor, but if there is no enzyme, you will not be able to have the catalysis and the metabolic pathway. So you will not find growth here, and you will find growth in the other parts. Yeah, you will find, if you will find growth in the other parts. Here, in this case, you don't find growth in this one and in this one. So you don't have growth here and you don't find growth here. Why? Because even you can have the precursors, there is no enzyme to produce growth. So the only part that you will find the growth is at the end, and you will have the metabolic pathway. So in this way, and you can read it in the textbook, you will be able to detect which one is the first one, which is the gene A, which one is the gene B, and this one is the gene C. So hopefully this was clear. And again, I think this topic you need to read it from the textbook, that so you can start understanding in more detail this concept. So here you find a different way to explain this topic, and maybe I can, in the test, I can give you this, yeah, and you will have to tell me the order of the genes, yeah, which one go first, which one go second, and which one will go third. And there are multiple exercises, and we will do solve some of those one in the classroom, so you understand this in more detail. Okay, so to finish this idea, based on this experiment, a gene is defined experimentally as a set of mutants alleles that make one complementation group. So for example, here you have 1, 2, and 3 genes, and here in this schematic you have here gene 1 and gene 2, here you have gene 1 and gene 2, same thing here. And if you see here, when you make a cross between mutant 1 and 2, what happened is that although you have a mutation here and you have a mutation here, they complement, because they are in different genes, and they are able to grow in a minimal media. Yeah, so you will find growth. On the other hand, when you have the mutation in the same gene, if you see here, although these two genes are working, and this means that in theory the fungus is going to grow. In the other case, both are not working. So as a result, as the mutations are in the same gene, there is no complementation, and there will be no growth of the fungus. So remember, if the cells of the organism is a mutant, the mutation failed to complement one another, the mutations will be located in the same gene. Okay, now we're going to move to another topic. Remember, this was to explain that there was a relation, there was a relationship between gene and enzymes, that in the long term, it was simplified to explain that there was a simple relationship between genotype and phenotype. In the following part of the class, I'm going to talk a little bit more about why genotype and the relationship between genotype and phenotype is not as simple. It's not that when you have one gene, you're going to have one phenotype. And I'm going to show you several examples, and to do that, I will also explain just a basic concept like mutation and other small things. Okay, so to finish this, we are going to talk about the central dogma of molecular biology. And I know that you studied this multiple times, so you know that the information will go from DNA to RNA to proteins and going in this direction. There are some exceptions. So, for example, some viruses and some enzymes can go from RNA to proteins, but we don't see, as far as we know, that there is something that can go from proteins to RNA. So remember, DNA is the informational molecule that acts through an intermediate, that is the RNA, to produce proteins. And of course, the process of transcription and translation uh, are very important. And the more complicated, the more complex the organism, the more steps we'll have. So in eukaryotic cells, you have, and we will study that in a lot of detail in the classroom, all these um, different tools that allow transcription and translation and that regulate it very well. So we will also talk about the different type of RNAs, and we will talk in much more details in the classroom about the RNA, the ribosomal RNA, this messenger RNA, and this transfer RNA, how they are formed, and we will talk about other type of RNA that they have appeared, because there is a lot of type of RNA that now are in charge of regulated transcription and translation. But main idea, DNA, you have transcription, then you form different type of RNA that then will go on to translation to synthesize proteins. So with this is the main idea that you need to remember for this chapter, and in the following chapters we will go into a lot of details about this. So in the class or in previous courses, you have studied mutations. Now in the lab, we are going to see the effect of mutation on fruit flies. Uh, Here, I just want that you remember the the main definitions of mutation and the different types. So remember that they could be a substitution. Yeah, that you have here. You can change in the nucleotides. And here is a point mutation. There are ones that are deletion and additions, and while the point mutations can have small changes, these can be they can alter the reading frame. So these mutations can produce much more dramatic effects. Okay, so this is a topic that also we have studied before, and I'm sure that you have heard a lot about the relationship between genes and environment. I just want to remind you that before the influence of genes was very high, everybody thought that genes was the determining factor in the past. Yeah, and the relationship between gene and phenotype was very simple. Now the role of the environment has been growing and growing and growing, and now uh, we know that there is a that both that in many traits genes and the environment are very important to understand the phenotype. So all these examples that we have with Mendel, they're beautiful, but in many cases, many traits, especially complex traits, are affected by multiple genetic and environmental factors, and I will show you that using fruit flies. And many genetic disorders, the severity depend on the combination of both. So, for example, cancer, although there are definitely oncogenes, genes that a mutation will lead to that cells start proliferating in an abnormal way, and when you start accumulating many mutations, cancer appear. Definitely, the environment play also a key role. So the level of stress that we have, the food that we eat, how many hours do you sleep, and also aging is all these factors are playing a key role. So it's the combination between both in many cases rather than only one of those. Okay, another phenomena that we will study in this class, that if you remember we studied it in genetic revolution, is the phenomena of epistasis and pleitropy. Again, the relationship between genotype and phenotype is not as simple. So one example was the role of the environment. Another example is the role of these two phenomena. So rather than finding that we have one gene and one enzyme, here I'm just going to show you that it could be more complicated than that. So for example, in the lab, we will show you that although I cannot show you that in humans and mutate some of my students will be a terrible idea. I will get fired, but I can have mutation of fruit flies. And you will see in the labs that when you have those flies, you will find that eyes can change. And I can show you other internal organs, or I can show you simple things like the wings, like the legs, like the bristles in the legs, that only one mutation will affect multiple traits. So there are these two phenomena that are called epistasis and Pleitropy that we will study in the lab, and we will study also in the class, in the lecture. So here, epistasis, you have multiple genes that will affect one trait, and we will study the Drosophila eye color to show you that multiple genes will form a pathway, will have proteins that will determine the eye color. So we will see flies of different eye colors, and how the interaction of those proteins will determine the eye color of a fruit fly. In addition to that, we will study the phenomena of pleiotropy, is the one that I showed before here in this schematic. And the idea is that you have one gene, and that gene determine multiple traits. Yeah, so as I said before, in addition to the environment, you can have other phenomena that make the relationship between genotype and phenotype not as simple as we think. So remember, one gene can affect more than one trait, and this phenomena is called pleiotropy. And any trait can be affected by more than one gene, as well as environment. And to finish with all these things of the chapter, I'm not going to talk about what is evolution. You already took so many classes about that, but this will be in your test. And just remember that genomics is the set of all the genes, while proteomics is the set of all the proteins, and metabolomics is all the set of metabolites. So now phylogenies are not only formed by genomes, but they can start getting more complex, and they can show us, if we combine all these different hierarchical levels, they can help us to understand more of the evolution. So it's not, before our phylogenies were mainly based on genes. Now they are formed by different genes and different genomes. We can go on try to understand better evolution if we understand all other hierarchical levels. And these are studies that we have already identified the entire proteins set of organisms like E. coli or yeast, and the same thing with E. coli. And we are finding a lot of interesting things that I'm not going to discuss right now, but these concepts will be in your test. Okay, so this was more a very summarized version of this chapter, because you studied, you know all those things. If you have any doubt, this is the time that you go to your textbook and review the topics that you have a doubt. I hope that you have a good day, and if you have any questions, just let me know.